All right, today we are diving deep, and I mean really deep, into the code of life itself. We're going to explore the incredible and sometimes heartbreaking link between our genes and our health. We'll unpack how our body's master instruction manual is written, how it's read, and, crucially, what happens when there's a typo.
You know, it all really comes down to this one question. It sounds kind of impossible, right? How can one misplaced letter out of billions have such a massive, life-altering impact? Well, today, we're going on a journey to find out, all the way from the microscopic world of molecules to the real-world effects on people's lives.
So before we can even begin to understand the errors, we have to get a handle on the original document. We need to look at the blueprint itself. And in the language of biology, that blueprint has a name we all know. DNA.
DNA, or for the science buffs, deoxyribonucleic acid, is without a doubt the master plan for every single living thing on this planet. I want you to think of it like the ultimate IKEA instruction manual, but instead of a bookshelf, it has every single piece of information needed to build and run, well, you. From the color of your eyes to how your heart beats.
And the way this information is stored is it's incredibly elegant. You've got this beautiful double helix structure with two strands twisting around each other, running in opposite directions. The instructions themselves are written in a simple four-letter chemical alphabet, and just like reading a sentence from left to right, our cells have a very specific direction they read this code in to make sense of it all.
Okay, so a blueprint is totally useless if no one can actually read it, right? It could be the most brilliant plan in the world, but if it's just sitting on a shelf, nothing gets built. That brings us to the next critical step. How does the cell take these instructions and turn them into stuff?
This whole process, sometimes called the central dogma of biology, happens in three main stages. First, replication. That's when the DNA blueprint gets copied, say for a new cell. Second, transcription. This is where a specific gene, a single instruction, gets photocopied into a temporary message called messenger RNA, or mRNA. And third, translation. This is where the cell's machinery, these little factories called ribosomes, read that mRNA photocopy and use it to build the final product. And what is that final product? Proteins.
If DNA is the blueprint, then proteins are the construction workers. They are the builders, the movers, the absolute workhorses of our cells. They do pretty much everything. They give our tissue structure, like the collagen in our skin. They carry oxygen in our blood, and they manage almost every chemical reaction that keeps us alive. They are what the DNA blueprint actually builds.
So, how does that mRNA message get translated? It's read in three-letter words called codons. Each codon tells the ribosome which specific amino acid to add to the chain, and amino acids are just the building blocks of proteins. So, for example, the codon UCA tells the machinery, hey, grab a serine and put it here. Simple enough. But then, look at UAA. It's different. It's not an amino acid at all. It's a stop sign. It literally tells the ribosome, okay, we're done. Stop building now.
And this is where we get to the heart of our story. If the DNA blueprint is an instruction manual, then a mutation is, quite simply, a typo. It's an error in that original text that can have huge, huge consequences when it's time for the cell to build something.
This right here shows the problem perfectly. On the left, you see the normal code, UCA, which correctly signals for the amino acid serine. The protein gets built just fine. But look what happens with just a single letter change. One tiny typo turns UCA into UAA. And what did we just learn UAA is? It's a premature stop sign. So instead of building a complete working protein, the cell just stops halfway through, and you're left with a shortened, completely useless protein fragment. This kind of typo is called a nonsense mutation.
And that's just one way the code can go wrong. There are others too. A missense mutation, for example, swaps one amino acid for another, which might be okay, or it might totally change the protein's shape and function. And then there's a frame shift mutation. This happens if you add or delete a letter. Think about it. It throws off the whole three-letter reading system, scrambling every single word that comes after it into complete gibberish.
Now, these aren't just abstract ideas or chemical errors happening in a petri dish. These typos have real, tangible, and often devastating consequences for people. So let's take a look at how one of these tiny flaws in the blueprint can lead to a complex human disease.
Our first example is osteogenesis imperfecta, which you might know as brittle bone disease. This is a classic textbook case of a disease caused by a defective blueprint for one single incredibly important protein, collagen. You know collagen. It's the main structural protein that makes up our bones, our skin, our teeth, basically the scaffolding of our body.
And because collagen is everywhere, the damage from this one faulty protein is widespread. It affects the entire body. There's actually a really great mnemonic for the symptoms: bite. B is for bones, which, without strong collagen, suffer multiple fractures. I is for the eyes, which can have blue sclera, that's the white part, because the connective tissue is so thin you can see the veins underneath. T is for teeth, which are often imperfect. And E is for ears, as abnormal bones in the middle ear can lead to hearing loss. All of that from just one typo in the collagen gene. It's astonishing.
Okay, next up, let's talk about Duchenne muscular dystrophy, or DMD. This is a truly devastating disease, and it's typically caused by a frame shift or a nonsense mutation, the exact types of typos we were just talking about. This particular typo is in the gene for a protein called dystrophin.
Dystrophin's job is to act like a kind of shock absorber, connecting the muscle fiber's internal skeleton to its outer membrane. Without it, the muscle cells are incredibly fragile and they just break down over time. And this leads to the classic signs of DMD: progressive muscle weakness, the Gower sign, where a child has to use their hands to walk up their own legs to stand, and enlarged calves that look muscular but are actually filled with scar tissue and fat.
For our final example, let's look at cystic fibrosis, or CF. This disease is caused by a mutation in a gene that builds a really specific kind of protein. It's a tiny channel, like a little gate, that lets chloride ions move in and out of our cells.
So what happens when this tiny cellular gate doesn't work? Well, the whole salt and water balance across the cell membranes gets thrown completely out of whack, and the result is this incredibly thick, sticky mucus. This mucus clogs up the lungs, which leads to chronic infections, and it blocks the pancreas, so digestive enzymes can't do their job. The problem is so fundamental that a key diagnostic tool is the sweat test, which actually measures abnormally high levels of salt or chloride in a person's sweat. Now, I know after hearing about the impact of these genetic typos, it's really easy to feel a bit discouraged. But the story absolutely does not end there, because by understanding the root cause right down to the letter, science and medicine are finding incredible new ways to fight back.
First off, we've gotten unbelievably good at reading the blueprint. Modern genetic sequencing lets us zoom in and find the exact typo causing a disease. And here's the hopeful part. While we can't always go in and fix the source code, at least not yet, we can develop some truly brilliant treatments to manage the downstream effects. For cystic fibrosis, there are therapies to help thin out that dangerous mucus. For osteogenesis imperfecta, certain drugs can help strengthen bones and lower the risk of fractures.
Which brings us to our final thought for today. We've gone all the way from a single chemical letter in our DNA to a complex human disease. We've seen how a tiny typo can become a tragedy, but we've also seen how understanding that typo is the first step toward treatment and hope. So the big question, the one that lies ahead for all of us, is this. As we get better and better at reading life's blueprint, how will we finally learn to correct its typos at the source?